Well, good afternoon, everybody, and welcome. My name is Tom Banchoff. I'm the Vice President for Global Engagement here at Georgetown and co-leader of our Global Health Initiative. And on behalf of the entire university and the initiative, I'm delighted to welcome you to our historic Riggs Library for a lecture by my colleague, Dr. Heidi Elmendorf, known to so many of you, to celebrate the fact that she's a finalist for the Robert Foster Cherry Award for Great Teaching. Heidi, we're looking forward to your talk on a wonderful topic, the moral relativism of microbes, to be or not to be virulent. We're very curious to hear what you have to say. Heidi has been a dynamic researcher and teacher here at Georgetown since 1999, where she serves as associate professor in our Department of Biology. Before joining our community, she studied at Princeton and Stanford and was a recipient of a 3-year postdoctoral fellowship at the National Institutes of Health. Today, you'll have the opportunity to learn more about Heidi's cutting-edge research on intestinal pathogens. But in my remarks, of course, I want to emphasize her impressive, really impressive contributions as a great teacher in our community. Heidi is passionate about her subject matter and a superb communicator, filling one of the largest lecture halls on campus for her Foundations of Biology course. How many of you here have taken that course? There we go. All right. Is that part of your assignment this semester? I don't think so. All right. She is, as you know, totally dedicated to the intellectual development of her students. Students, she engages you in the classroom, but also in the department where she long served as Director of Undergraduate Studies. Heidi, you really beautifully exemplify one of our core values here at Georgetown, cura personalis, or care for the whole person. And in that spirit, you also direct our innovative Regents Science Scholars Program, which expands opportunities for students from traditionally underserved communities interested in studying biomedical science. In recognition of Heidi's pioneering work with the Regents Science Scholars and her deep commitment to undergraduate education, she's recently taken on a new role here at the university as Senior Advisor to President DeGioia for Equity in Education. Now, let me list just some of the many awards Heidi has received as a teacher and educator to date. Here at Georgetown, she's collected a slew of accolades, including the Bunn Award for Faculty Excellence, the Dorothy Brown Award, and the Georgetown Academic Council Award, which she has won not once, but twice, in 2011 and 2017. Heidi's innovative teaching has also been recognized beyond Georgetown. In 2014, she was honored with the Carnegie Foundation for the Advancement of Teaching DC Professor of the Year Award. Heidi, we're proud of all your contributions to Georgetown as a star teacher and as an advocate for the highest quality undergraduate education. We're delighted, proud of the fact that you've been nominated as one of 3 finalists for one of the very few national teaching awards for higher education, the Robert Foster Cherry Award for Great Teaching, which is based at Baylor University. Congratulations on your nomination. We think it reflects well on us, on Georgetown's commitment to excellence in undergraduate education, and we look forward to your talk. So please join me in welcoming Dr. Heidi Elmendorf to the podium.
So I don't do podiums very well. So, so most of you have had me. Good afternoon.
Good afternoon.
Good afternoon.
Good afternoon.
Okay. So if you've had me, you know I can't start class without that because we do things as a community. So I want to thank Tom for the very generous introduction. And although this is technically a dress rehearsal for the talk that I'm giving next Monday down at Baylor University, where the panel will sit in judgment of it, this is, for me, actually, in some ways, the more meaningful of the talks, because this is a talk about who I am as a teacher, and this is the community in which I've learned to be a teacher. I've learned from my colleagues who are in this room, and I've learned, perhaps most importantly, from my students who are in this room. So this is my crowd, this is my family. And so this is my talk. So I have a confession to make, though. So the title of my talk, as Tom said, is The Moral Relativism of Microbes, to be or not to be virulent. But I want to peel back a little bit, so I am going to talk about this topic. But what I want to do today, and what I'll do in Baylor next week, is a little bit more of what I call the director's cut. And what I mean by that is this idea about providing not just the straightforward final film version, but the version that tells you a little bit more about the decisions that I made, that gives you a little bit of the voiceover. So there's a bit of a voiceover here at the beginning. Because when teachers teach, part of what we do is we talk about knowledge, we talk about how it is, we don't just tell students what we know, we talk about how it is that we come to know that, the epistemology. And we think a lot about pedagogy, our teaching practices, basically where it is that what we're talking about, the content that we're talking about, the ideas intersect with our audience. So how it is that we came to understand that material and how it is that we think that perhaps they can come to understand it, be provoked by it, be inspired by it. So when I found out about the cherry last spring, they asked me what I was going to talk about. And actually, unlike what Tom told you, I actually chose to talk about something that's not my area of scholarly expertise. So I am a microbiologist. I'm a particular flavor. I'm a parasitologist, and I study pathogens. But I've never really studied their pathogenesis per se. I've never studied their virulence, their ability to cause disease. I've actually never taught a class, a single class that was devoted to this topic. This is perhaps a counterintuitive choice for probably the most important talk of my entire professional career. But I'm nothing if not slightly renegade in my decisions. So, but I chose it on purpose because good scholars, excellent scholars, should be able to give excellent talks about their scholarship. But great teachers should be able to give talks about things that are at least a degree or two of separation away from their area of expertise. And they should be able to discover those ideas and figure out how it is that they're going to bring their students with them on that journey of discovery. So I've chosen to talk about something that I'm not an expert in. So if you came to hear the expert's version of microbial virulence, I'll do the it's not too late, we haven't yet closed the door back to the airport, off the airplane, and you can safely depart before we pull away and leave the airport. Okay, so with that disclaimer in mind, this particular topic, there are many things for the record that I'm not an expert in that I could have talked about. The list is perfectly endless. If you want to hear the rest of them, you could come to Rice 103 on Tuesday, Thursday, and Friday mornings at 8:30 all semester, because I talk about half of biology and claim to have expertise in that too. So feel free to join us. But the topic for today does have particular resonance for me. So one of the things is that I've come to this particular topic through my own journey. And that journey for me started back actually in my senior year of college. So these sorts of talks are rare, and you don't usually get to give them. So this for me is a chance to honor some of the people who helped me become who I am. And a couple of really important people in my senior year of college at Princeton, I had the good fortune to be taught by John Bonner. So Princeton didn't actually offer a microbiology course. Microbes had been discovered then, but there was not yet a course about it. So I took a private tutorial with John Bonner, and he taught me what I know about microbiology at the time. I also was really fortunate at the time to take a course from two outstanding cell biologists, from Pam Silva and Beth Luna. And they're the ones, for the first time, who helped me understand what was going on inside of a cell. I'm pretty sure my introbio professor had tried, but I did not take in most of what he said in that semester. And so they're the ones who finally convinced me about the beauty of cells. And then in my senior spring, I had the chance to work with a very important molecular evolutionary biologist who was at Princeton for a relatively brief period of time. But Marty Kreitman is the person who taught me about the beauty of looking at DNA, and looking at DNA as not just genetic material, but evolutionary material. So after their tutelage, I headed off to Stanford, as Tom said, and I studied parasites while I was there. And the year after I graduated, when I was teaching high school biology, which I did for 3 years, an important book came out. And this is a book by a man named Paul Ewald. So I'm going to try and show you not just faces of people important to me, but important scientists. Paul Ewald's an important evolutionary biologist. And in 1994, he published a book called The Evolution of Infectious Disease. And that book, in a sense, is the godfather of today's talk, because it's in that book that some of these ideas really first came to the forefront. So a lot of time has passed, and since 1994 to today, lots of things have informed this topic. So I did teach high school for 3 years. I taught at Phillips Andover Academy. And since then, I've been a Giardia biologist. That's kind of my research scholarly life. I live here in the biology department at Georgetown in our beautiful new building in Regents Hall. And I've had the good fortune to work not just with my colleagues in my department to learn about quality teaching and what that means, but also with Candles and the crowd of teachers on this campus that it brings in, and also the Carnegie Foundation for the Advancement of Teaching. So all of this has kind of brought me to a place that says, well, this was the day that I could finally go back to that book by Paul Ewald and say, what would it be like to actually talk about those ideas, but newly informed by everything that's come since then. So when I talk about things, I place a lot of value on the language, and I think it's really important to be precise in language and to understand its usage. So the title has a little bit of tongue-in-cheek, obviously, but the words mean something. And so the first word to pull out from the title that we can talk about is just the word microbes. So this talk is intended to be a talk for a general audience. Here, I'm seeing lots of science people in the room, but at Baylor, there'll be people who aren't scientists. And I did my own plant here, so my husband Randy Bass is in the room, which means I've got at least one English professor here kind of keeping me honest about my talk. But so microbes are single-celled organisms. That means that individually they're too small to be seen with the naked eye, and you have to use a microscope in order to see them. But collectively, if you allow them to grow up in small populations, then you can study them. So we have a petri dish here in the picture, where each of the little clumps on that petri dish are thousands or perhaps millions of microbes. And so there's bacteria and there's fungi all over that petri dish. So we can even see from this petri dish that there's a great deal of diversity. So if you say microbe, that doesn't mean one thing, that means many things. And we can gain a little bit of a better sense for that diversity. So on this slide, we see kind of the canonical tree of life. So there's 3 domains, 3 categories of life: bacteria, archaea, and the eukaryotes. So we're over here. This is where animals are, so we're at that far end. If we take a look on this sort of a schematic about where it is that we find microbes, we can see that we find microbes pretty much everywhere. So multicellular, non-microbial life is really quite limited. So we've got animals and fungi, some of which are also microbes, and plants. So really, we're in like the tiny little island, the outpost, if you will, of multicellularity. So by microbes, we mean a great deal of diversity of organisms, most of which never come into contact with a human or some other plant or animal multicellular host. And so if we want to talk about the ones then that are virulent, we're going to have to hone in on our discussion and peel off that word virulent, and then we'll be able to look at just a few of the microbes on that particular tree of life. So I did what I usually do when I want to make sure I'm really precise about the definition of a word. So I turned to Merriam-Webster online. It's my favorite online dictionary. Virulent doesn't get you quite as far because it's an adjective, so I used virulence for this. And it turns up these definitions. The most relevant one is the bottom line there. So virulence is the relative capacity of a pathogen to overcome body defenses. Basically, it's the ability of a microbe to cause disease. One of the new features of Merriam-Webster, in case you're just like a little dictionary fiend like I am, is that they have a new feature called the time traveler, which tells you when a word first came into at least recorded existence. And so virulence comes from 1617. It was interesting. So at that particular time, there was an era in which plagues, epidemics were routinely sweeping through cities in Europe and London. We were discovering, rediscovering, just like newly encountering whatever the new world. And we brought with us diseases to that world as well, and so we were bringing epidemics, and they were sweeping through populations. It's at that point in time, that exact same year, indeed, that quarantine and public health also came into usage, right? So that you could tell what folks were thinking about. The second favorite thing you do, of course, with a word is you go and you see what Wikipedia has to say about it. So if you go to Wikipedia and you type in virulence and you take that entire page and feed it into a word cloud generator, you get this. And so here's virulence down at the bottom. It's obviously said a lot on that page. But some of the other large words here tell us a bit of the story we're going to encounter. So it's a story about the microbe. And the microbes that we'll talk about today aren't all bacteria, but many of them are. It's about their encounter with the organism that they're going to live in, perhaps cause damage to, and that's their host. And we can begin to see other words up here. We're going to talk about how they get back and forth from one to another. It's the little gold word down here, transmission. We'll see some ideas about viruses and things like that as well. So this leaves us then just with one word left, or one phrase left, and that's to try and define moral relativism. So I pulled the same trick that I did before, which is I went ahead and I typed it into Merriam-Webster, and this is what comes out the far side. So this was a grim discovery, because this was already highly committed to being the title. It was like all over the website down at Baylor. And then I thought, well, that's okay. Merriam-Webster doesn't have phrases, it just has solo words, but that's super not true either, actually. It has all sorts of phrases in it. It was terrible. So then I took this possibly, though, as a sign from on high, because, of course, in theological circles, morality is not relative. As a matter of fact, Tom Banshoff, in a book that he edited, there's parts of that book that talk about the fact that you can't talk about morality as being relative. Morality just is. It's not up to you to get to decide. So possibly here at Georgetown, at a Catholic and Jesuit school, and down at Baylor as well, which is a religious institution, maybe I really just should have picked a different title. But it was too late. So the silver lining to the cloud is that I'm not talking about moral relativity in the abstract. I'm talking about the moral relativism of microbes. And what I mean by that, I'll give you a specific example, and we'll keep talking about it today. So my specific example is this one. So it's a picture from a Scientific American article from about 5 years ago. And it's pointing out that there's a number of microbes that live in your intestinal tract. And the top one here, that's pointing to your stomach, the microbe they describe is Helicobacter pylori. So Helicobacter pylori is the causative agent of ulcers. So that earned somebody a Nobel Prize. And for the record, he got the Nobel Prize because he drank Helicobacter pylori, gave himself ulcers, and people will do anything for a Nobel Prize. So that makes them bad. So from a moral perspective, these are just bad microbes. But on the other hand, more recent research has helped us understand that Helicobacter pylori's presence also helps your body to properly regulate acid levels in your stomach, and also to properly regulate hormones that are responsible for controlling appetite. So that sounds good. So I don't know, so is it bad or is it good? More complicated, about half the people in the world have Helicobacter pylori in their intestinal tract. Those numbers are a little bit lower in the US because we're super antimicrobial everything. So we've actually done a pretty good job, bad job, effective job of weeding out some of your normal microbiota. But around the world, it's quite common. And of those 50%, at most about 20% will ever develop ulcers or even ulcer-like symptoms. So not like full-blown ulcers, but maybe the tendrils of one. So they do bad things and good things, and in people who have them, most of the time they don't do anything evil, and some of those people, they'll never do anything bad, and in some of them, only occasionally upon provocation will they do something bad. So that's moral relativism from a microbial perspective. Okay, so So here's another confession. So I had a talk written, and then as I was practicing it at about 10:30 last night, I realized I'd written the wrong talk. I'd realized that I'd written a talk as though I was trying to be an expert in this, and I was trying to give a seminar. And that's not why I picked this topic. I picked this topic because it's something I'm discovering, and because I'm good but not an expert at it. And I'd written the wrong talk. So at 10:30 last night, I tossed about 2/3 of the slides. I inverted the remaining third, and then I didn't get a lot of sleep between then and now. And I wrote the talk I think that I was supposed to have written. But occasionally, at that moment, it was like, dang, what clip art can I pull out and put into it? So that's this. Okay, so we should talk though, right? So that Helicobacter pylori story, what I told you there, some of that information, the ulcer information is from the 80s, but the newer work on appetite and acid levels, that's from the 21st century. So how did we get here? So what does the journey look like? So let's take a little bit of a historical perspective for this interaction between microbes and their host. And the story starts like most good biology stories start, back in the early 1700s with Antony Van Leeuwenhoek. He is justifiably famous for being the individual who both rocked a very nice-looking wig and, in his free time, built microscopes. So this wasn't his job, this was his hobby. And this is one of his rudimentary microscopes. You'd put your sample on the head of that pin, and there's a little bit of a lens behind it that you can go ahead and look at it. And so he quickly realized that no matter what he sampled, within it, there were clearly alive objects. And so he drew them, and he knew that they were alive, not just because he could draw shapes, but because some of these objects, as indicated by the dotted line here, were moving. And so he discovered the microbial world. He discovered it in the world around him. He discovered it on the surface of his skin and the scrapings of his teeth. And because I do my research on Giardia lamblia, most importantly, he discovered it by looking at other output. And is the first to actually have described Giardia. So at his point in time, in the changing view of microbes, it's just like, woo, gosh, they exist. They are microbes. 150 or so years later, in the late 1800s, microbes started to get a bad rap. So largely through the work of individuals like Louis Pasteur, who came up with the germ theory of infection, profoundly important, right? That this idea that disease doesn't come from bad air or just bad luck or the phase of the moon, but that there are actual things that are causative agents of that disease. And his work was, in a sense, codified by Robert Koch. And so Robert Koch did his work in the late 1800s, earned a Nobel Prize for his work, not the least of which is pinning down that a very particular bacterium is responsible for causing tuberculosis, for example. But he did that. He made that correlation between microbe and disease through the series of what have become to be known as Koch's postulates. And so a shortened version of them are shown here. But it's this idea, though, that if you look at sick people, you should be able to find a microbe, let's say a bacteria within them, that should not be present within healthy people. If you take the microbe out of the sick people and grow it up and make sure it's pure and put it back into a healthy person, you should be able to make them sick. So this was the Helicobacter pylori causes ulcer experiment. He did Koch's postulates, but on himself. Right? But this is a very simplistic notion. Microbes are bad. And if you have a microbe, it causes a disease, and it always causes that disease. So that's not true. Because our culture is a big fan of yogurts. And it turns out it doesn't matter what yogurt you have. You can have kids Go-gurt in a tube. You can be a fan of Greek yogurt or Icelandic skyr, as I am, or it doesn't really matter. All the yogurts are made as the products of the microbes that are in them. So they say they're live cultures. And we actually tout their benefits, right? So Activia, I always use this one because it's got such a stupid name, but Activia is a probiotic yogurt. Right? So this is probiotic. This means it's got life in it, and it's good for your life. And this is an actual legitimate ad. It contains an exclusive culture, Bifidus regularis. So Bifidobacterium is a well-known inhabitant of your intestinal tract, and so that's what this is. And so, so the world, our view changes a little bit. So, a few microbes are good. These particular microbes are good. But then in the 21st century in particular, our notion of what good microbes are has greatly expanded. So work now is done not just looking at individual microbes, but taking entire communities and trying to find out what all microbes are present in those communities. And so some of that work is focused, because we are innately a selfish species, on the human microbiome. So what all microbes are part of us. This is work that's involved hundreds of scientists over a couple of decades now, but the results of it turn up like something here on the right. So this is a chart showing front and back of an individual and all of the different locations on their body where they tried to sample to see what microbes are present. And different microbes have different colors, so each little color wheel at each of these locations indicates the identity and some of the diversity of the microbes that are found. Good party, cocktail conversation thing is to be able to note that if you were to take all of the cells in your body and count them, at most 50% of them are human cells, and the other 50%, maybe as much as 3 out of every 4, are microbial cells. They're not just there, but we know that they're important. If you remove them, and you don't do this to people, but to animals in the laboratory, those animals are less healthy. They find it harder to digest food. Their immune systems do not develop as well. Their brains do not develop as well. So these microbes are inherently not just on us and in us, but they're actually an essential part of us. So the new mantra out there is that microbes are great. Many, maybe the vast majority of them, are really good microbes. So as a microbiologist, that's good. It's kind of like the redemption of my entire field. And then if you take a look at the very start of Paul Ewald's book, so he says in his preface, and this is, I just took a screen grab, right? So this is why this book. And he's got quotes from 3 very famous biologists. So from René Dubos, he says, given enough time, a state of peaceful coexistence eventually becomes established between any host and parasite. Disease, Lewis Thomas, disease usually represents the inconclusive negotiations for symbiosis, a biological misinterpretation of borders. And Paul Hoprich's ideal of parasitism is actually commensalism. And commensal microbes are like the ones that were all over our bodies. Those are the ones that we think of as the good guys, the ones that are just part and parcel of who we are. Okay. So 3 of the most preeminent scientists from the late 20th century all agree. Microbes, they're just good, right? And we've just been mistaken. The rare exception is when you get disease. But that can't be true, right? Because Helicobacter pylori is both good for you and can be supremely dangerous. People die from ulcers. Right? So I don't think it's that it's good or it's bad, it's that it's complicated. And that's moral relativism again. Okay, so if we're thinking about moral relativism, how relative, then, shall we say, are the microbial morals of virulence? Like, just how far can you stretch this? What are some examples of this? Okay, so let's do a revision of this. Hey, once you find clip art and it's 2:30 in the morning, you just stick with that clip art. So let's talk about microbial virulence, a present-day understanding. So this is the really, I thought bullets were a good idea, but we'll go through it slowly here. So microbes can be good, commensal, bad, a pathogen, or neutral. Even bad microbes, even the pathogens, can fall very different points on the spectrum. They can cause subtle disease, not a big deal, common cold for most people. They can kill you. They can be Ebola. What determines this status? What determines how dangerous they are? Well, it's a little bit of them and a little bit of you. So microbes, it depends in part on their numbers, and we'll see some of these examples in coming slides. It depends on how fast they can reproduce, not just how many of them there are, but how quickly that population's growing. It depends on their genetic potential. So it depends on what genes they have and what those genes allow them to do, what blueprint of instructions they give them. But it also depends on you. It depends on your response to them. It depends on how much you fight back. It depends on how healthy you are. It depends on your age. It depends on your nutritional status. It depends on your gender. It depends on all sorts of things, your hormones. Many features of you feature into your dynamic with the microbes. Your behavior, how it is that you socialize with one another, the sorts of social networks that you form will be important. And that's important because part of what matters here is not just the microbe in isolation or the host in isolation or the intersection. So it matters how long that microbe and host have been interacting with one another. And I don't mean like days, like the person in Foundations in Bio sneezed on you yesterday. I mean, have they been interacting? Have they been a pathogen or a microbe associated with humans for 1,000 years? Or have they been associated for 500,000 years, right? What's the time frame of that interaction? How do they get from host to host? If a microbe only manages to live in you and you die, the microbe dies with you. So by definition, part of what we have to be talking about is how microbes get from one host to the next. And that's the idea of this transmission. So let's play out a few examples. And I think if you're squeamish, this is kind of the only squeamish slide. So we'll get it out of the way early. OK, so I want to talk about, and I pulled out really a few out of what could be many. We're starting with the bacterium. So Staphylococcus aureus is a bacterium. It's up here, this little kind of cluster of what looks like grapes up in the top left are individual bacteria. Each little ball is an individual bacterium. Staph aureus is a normal part of most people's skin. Even the most dangerous form of Staph aureus, the MRSA, the methicillin-resistant strain of Staph aureus, is still found on a relatively high percentage of populations. People in this room definitively have it on their skin. And you probably your skin looks like this panel on the left, which all's fine and good. But you also can get a Staph aureus infection that looks like the middle one with a little bit of an infection, or you can get a wound, an open sore that's like on the right there. Right? And those later stages, so for many people, you have it and you'll never know it. For others, that wound is just the beginning. There'll be over 100,000 hospitalizations this year in the US alone. And about half of those individuals hospitalized with MRSA infections will die. So this guy's pretty far off on the very high mortality end of things. But some of you here have it, and you'll be just fine. So that's one of those interesting dynamics. One of the things that we know, so how does Staph aureus do this? One of the things that we know, and there are many things that are interacting, your health, people who are elderly or in poor health or have weakened immune systems will get sick and die from MRSA well before other individuals. But part of it's about the bacteria as well. So one of the things that Staph aureus and other bacteria, other organisms can do is they can keep a head count. So they do something called quorum sensing. They can figure out based on based on chemicals that they're each making, how large the population size is. And they'll change their behavior based on the population size. If there aren't many of them, they know they're outnumbered by your body and your defense system, your immune system, and they'll stay quiet. If their count gets to be high enough, number matters, replication rate, reproduction rate matters, then they'll sense that and they'll start turning on genes that make them more virulent. So virtually all staph has the ability to be virulent, but it's a question of whether they're going to use that as a weapon against you or not. And they'll do so based on a head count. Not all microbes are bacterial. Some of them are protozoan parasites. That means they're eukaryotic cells like our cells. I have two favorites in life. I have one that I studied for my graduate work, and that's malaria, and I have Giardia that I work on now. And so Plasmodium, there are many different species of Plasmodium. There's about 5 that cause disease in people, and the 2 most common and most important are Plasmodium falciparum and Plasmodium vivax. I put these up here side by side because if you look at these little sketches down below, and these are just sketches, but they're pretty accurate of what the parasite looks like growing in red blood cells, they're kind of hard to tell the difference between them. If you're like, I don't know, that looks like little purple specks and like the kind of pale reddish colored red blood cells, well done. That's about what they look like. You can look at the life cycle, so you can go to the Centers for Disease Control website and pull out a life cycle. And it really doesn't matter which one you pull out because they look pretty much identical. Now, if there are malaria people in the room, yes, I know. They're not actually exactly identical, but they're really darn close. But Plasmodium falciparum causes about 92% of all cases of malaria in the world and is responsible for an equally high percentage of deaths. Right? So it has a higher mortality rate, and it just has a lot more cases. So falciparum is clearly the more dangerous one, but they're very closely related. That's one version of moral relativism of virulence. But another version becomes equally important. So the vast majority of individuals who die from malaria are small children under the age of 5. For a number of reasons, kids are more susceptible to all diseases pretty much, and they're particularly susceptible to malaria. If you survive beyond that age and you make it into adolescence, your chances of surviving from malaria, from subsequent infections, is actually quite good. You've developed what's called partial immunity, so you're pretty safe. Unless you're a woman and you get pregnant, in which case all bets are off again. So the immunity that you've developed is against versions of the parasite that not pregnant people see. When you're pregnant as a woman, you have new body compartments that you did not have before. And those body compartments provide a new habitat for the parasite. The parasite makes use of those new habitats, and new versions of the parasite appear that aren't found otherwise. You have no defenses against them. And so once again, you can get sick and die from it, and your developing fetus can be affected. Low birth weights, miscarriages are common. Same parasite can be infecting your husband, could have been infecting you a few months ago before you were pregnant, and you'd be fine. But now that you're pregnant, the rules have changed. So that's moral relativism of microbes. We can take a look at a virus. I know viruses aren't technically microbes because they're not living things, I know that, but everybody always wants to see a virus. So we can look at something like influenza virus. Most years, what I'll say is typical. So this is a virus down here. It's like proteins and nucleic acid, right? So the typical year over here, there's somewhere between 250,000 to 500,000 deaths around the world, and it's about a 1% mortality rate. So of the people who get sick, about 1% die, and that's mostly among the elderly or those who are immunocompromised. People in this room in general, a bunch of you will get the flu because you will have not gotten your flu shot like you were supposed to. And you'll need extensions on all of your coursework because you'll feel terrible, but you'll recover just fine from it. Okay? But then there are years like the 1918 Spanish flu, when around the world it's estimated that 20 to 50 million, and every time they go back and redo the estimates, it seems like we just bump up another 10 million. So most estimates now are coming in closer to the 50 million mark. Individuals died from influenza and its associated diseases. So that was about a 5 to 10% mortality rate. So many more people got infected and a much higher number died. So this is a difference, and I should say that the people who died here, it was across different demographics. It wasn't the elderly. It was actually the people in the prime of their life. This room would have been a hotbed for Spanish flu. So a completely different profile, a different level of danger, and a different target audience. So that's a moral relativism. We've got something in the wings there. The avian influenza that I point to at the bottom is one that gets passed between, shockingly, birds. It can go from birds to humans, and if humans get it, they actually get quite sick, and the mortality rate is quite high. So it's got an over 30% mortality when a human picks up avian influenza. The thing that saves us at the minute is, I said, that transmission, the ability to get from host to host, is very important. And thus far, humans have been unable to transmit their case of avian influenza to other people. So that's our saving grace. But the pathogen itself is quite dangerous. I'm going to try and keep careful track of time here. So I'm going to skip a couple of slides, but I promised this slide. So I'm going to tell one more story, and it's a story also about moral relativism in parasites, and I'm a parasitologist, so if I'm going to pick one more example, it'll be a parasite example. And so Toxoplasma gondii is a parasite that's actually pretty closely related to malaria, evolutionarily. Its normal cycle is between cat and mice. This is not actually a joke, it really is between cat and mice. It develops in mice, and it actually usually makes its way, the parasite, to the brain of the mouse and sets up an infection in the brain. Cats come along and will kill and eat the mouse. And if you've ever had a cat, you may notice that one of the first things that cats do is they actually dissect the head off of the mouse and then eat the brain. Right? So, and so they pick up the parasite from that. The parasite then has sex in the intestine of the cat, comes out in the feces. Mice pick it up from the feces of cat. That step has never fully been explained to me, what mice are doing in the feces of cats. But they pick it up, and the cycle continues around and around and around. Okay, so this is probably not terribly good for this little loop. But the problem comes that those parasites can occasionally make their way quite aberrantly into other animals, and they can set up infections in livestock and in people. And in those individuals, it basically thinks we're a mouse. So it does not, as horrifying as it would be, it doesn't have sex in our intestinal tract. That seems gross enough. But it instead sets up an infection and makes its way to your brain and sets up residence in your brain. Around the world, whoops. I don't know where the number is. Around the world, about 1/3 of the human population is infected with Toxoplasma gondii right now. That means about 1/3 of the people in the world have Toxoplasma gondii in their brains. Most of those people will never know it, and they'll be just fine. For some of those people whose immune systems get compromised, you'll begin to see those parasites growing in the brain. You'll begin to see the host itself trying to fight back a little bit, and you get these lesions that show up. This also, right, so there its danger depends on your level of defense. It also, though, once again depends. So pregnant women are just picked on by pathogens, it turns out. So if the first time you get a Toxoplasma infection is in the first trimester of pregnancy, that Toxoplasma infection will not just set up residence in you, but it will also harm the developing fetus in the placenta. And so it's known to be a causative agent of birth defects and, again, for miscarriages. So the host matters and the parasite matters. I show this as an example because of one quirky, and I like this story because we don't really yet know the answer to it. There's evidence for, and there's a little evidence against, and there's a bunch of evidence in the middle. But the hypothesis is this, and a number of studies seem to suggest that infection in the brain with Toxoplasma, even when it seemingly has no effect on your health, it's in your brain, and it's altering your neuron function. It's altering your behavior. There have been studies that are pretty good definitive studies with lab animals that lab rodents lose their fear of cat urine. They know to stay away from cats, but rodents who are infected with Toxoplasma aren't as scared as they're supposed to be of markings, the scent of a cat. They're risk takers, and it doesn't usually go well for them. There's a lot of questions about whether that's something unique, because that cycle is exceedingly well developed. This cycle has been in existence for a long time. The one in us is relatively newer. So maybe something different is going on in rodent brains, or maybe people who are infected with Toxoplasma also are risk takers, or are subject to other neurological consequences, mental health issues or cognition issues. Depending on what month it is and what journal you pick up, you'll find articles that are arguing for or against. That usually means that the story's somewhere in the middle, and it's not really quite as dramatic as some of the early publications. The story first came out about 5 years ago. There was a big flurry of papers about 5 years ago. But it's complicated. So here we have something that's not just causing disease, but is also potentially altering your behavior, and doing so to enhance its own spread. Okay? So that's moral relativism. So I did a little bit of a disservice before when I gave this quote from the start of Paul Ewald's book, because what I put in the front was the other scientists he quoted. But Paul himself responded with this paragraph. To all the people who assured us that it's all about like getting on well with others, he writes, "Few ideas have been so ingrained in the literature of medicine and parasitology as the idea that parasites should evolve toward benign coexistence with their hosts. Few ideas in science have been so widely accepted with so little evidence." And few ideas are so at odds with the fundamental principles on which they are supposedly based, with such a great potential for missed opportunity. The proponents of this idea rarely trace it to fundamental evolutionary principles. And when they do, they reveal a misunderstanding the most basic evolutionary process, natural selection. So that's a little bit of an indictment about some pretty famous biologists. So let's see what he's talking about then. Guys, it really was. It was like 3 o'clock in the morning at this point. Okay, so let's look at this then, not from a like here and now, like a couple of examples that are out there, but let's look at it from an evolutionary perspective. How did microbes get to have the relationship with the host that I was just describing? Right? How did we get here from there? So we're going to talk a little bit about evolution and what I call the arms race here. So if you're going to talk about evolution, it's actually mandatory. They throw you out of all the biological societies you belong to if you don't put a picture of Charles Darwin up on the board. And so there's Darwin, and there's very importantly the very first phylogeny, evolutionary tree, from his notebook. This is like the admonition to all the students in the room. Make sure you keep good notebooks because you never know who's going to be like flashing it up on a screen 150 years later. So this is a page from his notebook. You'll see we encourage people to like go with your uncertainty in my class. It's all about embracing your uncertainty. And he puts at the very top of this, he says, I think. Like, yes, dude, you thought well, right? So I also picked a picture of Darwin as he looked when he was doing the work, not as he looked like the week before he died. I think I had the impression that every scientist was like 90 years old when they did their discoveries, and that's not true. Most scientists are actually pretty young. I don't know what we do with the rest of our lives, but like everyone's Nobel Prize winning work is from when they're young. So this is Darwin when he's young and doing all of this work. And the basic tenets are this idea that you need variation in a population. You need genetic variation. You need Plasmodium falciparum and Plasmodium vivax. You need variation within those. All those parasites can't be identical. You need variation in host. You need the genetic differences that we see in this room around us. You need, at least some of the time, you need selection. So you need forces, survival of the fittest. You need forces that are going to select for some variants being better off than others. And I will say what we mean by that selection is not selection in the here and now, but is selection for like your survival, but is selection for the ability of that variation, your genes, to get moved forward. Right? That's what's going to be critical. If you live but don't reproduce, then all of your variation dies with you. And that's not an evolutionary success. So this is about variation and selection on that variation that allows genes to move forward. And that's really what we talk about when we mean evolution. You don't always need selection. It turns out there's a whole bunch of qualities about you. You can just think about some of your traits that have nothing to do with your reproductive abilities. And they're just kind of along for the ride. But there certainly is some selection taking place. These ideas have been echoed by many scientists. One of my favorite is Lynn Margulis. Did I get it right, Manus? Close enough. I apparently, and many other people, always mispronounce her name. Who was talking about these similar ideas, that basically all organisms, the kind of the quote I pulled from this is, all organisms today are equally evolved. So as we look at interactions from today, even when we look at microbes, which seem simple and from like way back in time, the microbes that are in you today are as evolved as you are. Right? So this isn't about evolution of something else. This is about you and your microbes are continually co-evolving. There's a particular version of this coevolution that matters. And the version of the coevolution that matters is this idea that something called the Red Queen hypothesis, which is that if you and the organisms that you're interacting with are continually evolving, some of that evolution is just to stay in the same place. And so that it gets its name from Lewis Carroll's Through the Looking Glass. But this idea that if you want to get somewhere else, you have to run twice as fast. It takes all the running you can do to keep in the same place. This idea was both developed intellectually and popularized by a number of scientists. Maybe the most famous is W.D. Hamilton, who published a very famous paper on sexual reproduction as an adaptation to resist parasites. So the goal then, if we reframe this, and we try to look at it not as a goal in the abstract about like just virulence, but the goal is an evolutionary one, which is then a goal of survival. And survival not of you, but a survival of your genes. Right? Then we're talking about reproduction, and we're talking about the continuance of the genes, of fitness. Or, as Gary Larson would say, and as amoebas, you'll have no problems recruiting other sales reps. Just keep dividing and selling, dividing and selling. Right? So this is evolutionary success. So what that means then, if we reframe this, this means that virulence, when we look at virulence, what's the right amount of virulence? How virulent should you be? Where do your morals fall on this spectrum? The right virulence is the one that helps your genes move forward. And that's going to be different at different times in different places in different situations. So that's where the relativism comes in. There are lots of things that microbes can do. So the left half of this is all about microbes. There's lots of things that microbes can do to be virulent. They can cause damage to the host by trying to just get in and set up home. They can compete with you for nutrients. They can outcompete those microbes that are just part and parcel of you, that are good for you, and cause them to leave. They can, this is the polite way of putting things, they can induce their own expulsion. I study intestinal pathogens, but this could be about all the people who are sneezing on you in lecture next to you. That's often, that tickle in the throat is often caused by the microbes themselves. You exacerbate microbial virulence. So in trying to defend yourself, you cause a lot of damage. Most damage in case of an infection isn't caused by the pathogen, it's caused by your immune system. And you can also try to expel the microbe. So if you want to damage the host to establish an infection, here's a good example. So this is a cell. And what's inside of it, all these little dark things, are actually bacteria. So these are bacteria inside of a human cell, or inside of a mammalian cell. These guys are Rickettsia bacteria. When they're done eating up the cell for the source of nutrients, they'll burst it open, destroy it, go forth. Each of them will set up home in a new cell, and so on and so forth. So they're using you not to damage you. You're just a food source for them. But they're going to inflict a lot of damage along the way. You're in constant competition with your microbes for nutrients. Everything in your body's trying just to survive. One of the really common ones is shown on this slide. One of the limiting agents in your body for your own growth, and therefore for microbial growth, is iron. Iron turns out to be very important in the functioning of your metabolism. And this has nothing to do with whether you're anemic or not. Your body regulates it such that it's only ever marginally available. So set up competition. You and your microbes are constantly trying to out-eat each other when it comes to iron. It can get rid of the good guys. So one of the most common infections in hospitals is Clostridium difficile, C. diff. C. diff, one of the dangerous things that it does is it's not just that it infects you, is that it clears out, disrupts your normal microbiota. It induces expulsion. So Vibrio cholera causes the disease cholera. Individuals can lose up to 20 liters of body fluid a day in diarrhea. And that's set up by the action of the bacteria that's causing the cells in your intestinal tract to release salts and waters. They're trying to flush themselves forward. Why? Because the game's transmission. And if they expel you in a huge volume of fluid, the odds are much higher that you're going to get into the water supply and be able to infect someone else downstream. But remember, and individual microbes can do lots and lots of these different mechanisms. So it's not like everybody's got a one-trick, they're not a one-trick pony. There's lots of different mechanisms within an individual microbe for causing disease. But you can cause disease too. You can damage yourself further. You can exacerbate virulence. So a streptococcus infection in the throat, strep throat, very prominent in kids, that bacteria does damage. It eats its way into your body tissue. It does a lot of the things we talked about. But the nasty symptoms of this, these swollen tonsils that are leaking this exudate, this wasn't caused by the bacteria. This is your immune system. This is your innate immune system trying to fight back. And it's the one throwing hand grenades, in effect, all over the place, hoping it kills off the bacteria, but damaging an awful lot of your body tissue along the way. So whose fault is that? But this is war then. Right? This is escalating war, because if you fight back, then the microbe fights back again, because this is about selection. So if the microbe can't overcome your defenses, then you win. So you'll select for microbes that can overcome defenses. But if the microbe can just kill you off too quickly, then everybody loses. So it selects for people who can fight back a little bit. So the who's winning gets complicated. And it gets complicated in a couple of different ways. One important way it gets complicated is about the rates of evolution. The hosts typically have very long lifespans. It takes us on average 20-something years to replicate. It takes microbes in you something on the order of bacteria of 30 minutes to replicate. Replication is when most evolution happens. So microbes evolve much faster than their hosts do, and that skews the power balance of this virulence game. I'm paying careful attention to time, and it's telling me that I should move on to my very last slide. So the last slide is about that this also, though, just doesn't matter in some abstract sense. This matters in our everyday practice. So it turns out that hospitals and the settings in which we treat people who are infected with virulent microbes plays its own role in this game. We put people close to each other in hospitals. Most Clostridium difficile infections are what are called nosocomial. That means they're hospital-acquired infections. People get them after they go to the hospital. They don't go to the hospital for treatment of them. They get them once they're there. And so we're changing barriers to infection and the cost of transmission. That will alter the virulence game. All of this work is coming into play as we think about drug and vaccine design. So currently, most drugs and vaccines are designed to completely eliminate the microbe. But should we be doing that? Most microbes, in very low numbers, don't really pose a threat at all. And indeed, most of them, at those numbers, we'd call commensal. We'd think they're there to help us. So maybe we're playing the game of medicine wrong. Maybe what we want to do is help keep them in check, help reduce transmission, but play along with the evolutionary game about the moral relativism of virulence. With approximately 30 seconds to spare, thank you very much, ladies and gentlemen. Students don't clap for me after class. So down the hall is the convocation with President DeGioia. If you're a faculty member, you should go to that thing quickly. But I'm actually happy to stay around and answer questions for individuals. And once again, I'm really deeply grateful for people's presence here. This was a privilege to get to give this talk before my family. So thank you.